Hi, this is Henry Mahncke from the Posit Science team. Today we're going to be talking about the basic science of brain plasticity and how we can apply that science to build brain training exercises that actually improve cognitive performance. Now, this basic research is really near and dear to our hearts here at Posit Science. Our co-founder, Dr. Mahncke, is actually one of the people who discovered that the adult brain was plastic back in the day. And of course, that discovery was incredibly controversial back then. But now it's a part of every standard neuroscience textbook. And the reason we're talking about brain plasticity today is because the idea of brain plasticity is core to every single exercise that we build here at Posit Science, because these exercises are quite literally designed to improve the speed and accuracy of information processing in the brain and to strengthen core neuromodulatory function, all through the power of brain plasticity. So let's dive in. How do we build brain training programs that work? Well, let's start off with what people want. What people want is to notice the difference in their lives. They want to have sharp cognitive performance at work. They want to have a brain that can support their social lives, all the things that they enjoy in their leisure activities. And of course, fundamentally, they want to have healthy brain function. Now, it turns out that what science has shown us through research over the past 30 or 40 years now is that real-world function, in fact, rests on common cognitive functions, things like attention, language, learning and memory, and visual, spatial, and auditory function. And of course, these are all areas that we see changes in over the aging process, or deficits in common neurological and psychiatric indications. Now, it turns out that these cognitive skills themselves rest on some basic aspects of brain function, things like the speed of information processing in the brain, the accuracy of information processing in the brain, the integrity of neuromodulatory function, systems like acetylcholine, dopamine, norepinephrine, and serotonin. Now, the good news, as we'll talk about in the next few slides, is that all of these basic aspects of brain function are plastic. We can improve them through brain plasticity. And our goal then is that by improving these basic aspects of brain function, we can see improvements in cognitive function, and then by improving cognitive function, we can see improvements in real-world function and quality of life. So what do we mean by brain plasticity? Well, fundamentally, brain plasticity means the brain's ability to adaptively reorganize itself throughout life. And brain plasticity is the biological correlate of learning and memory, and it includes things like structural changes in the brain, functional changes in the brain, and even chemical changes in the brain. Now, scientists' understanding of how the brain works has undergone enormous change over the past few decades. If you were to ask any neuroscientist back in 1970 how the brain works, they would have said, well, the brain's like a computer chip inside your head. It's made up of little electrical wires. They process information, and that's how you think, and that's how you act, and that's how you remember. And of course, because the brain's like a computer chip, people thought of the brain as hardwired. Fundamentally, once the wiring was laid down, after maybe an early critical period of childhood, you had the brain you were going to have. And in fact, all that was going to happen to your brain after that is it was going to wear out over time, or it might be damaged. But in both cases, those changes, those deficits, were fundamentally irrecoverable. It's important to say that viewpoint on how the brain works has undergone a complete revolution now, and every neuroscientist would now agree that the brain is soft-wired, or plastic. And what that means is the brain is constantly adaptively changing itself in response to what we ask it to do. And in fact, the brain is plastic when we're kids, it's plastic when we're adults, and it's plastic as we get old. And it's plastic when we're healthy, and it's plastic if we are sick in various clinical indications. In all of those situations, the brain can change itself. Okay, so let's talk about how researchers discovered brain plasticity. This was work that was originally led in the laboratory of Dr. Mahncke, the co-founder of Posit Science, and these experiments were led by Dr. Bill Jenkins. What we're looking at here is a monkey who's been trained to reach out and do a simple task. He simply has to put his fingers down on the surface of this rotating disc, which is like a record player. And the question these researchers asked was, well, if a monkey's trained to do this, and the monkey does it day after day for a few weeks, does anything change in the organization of the somatosensory cortex, the part of the monkey's brain that relates to touch? Now, if you believe the brain is hardwired, you would think, well, no, nothing changes. So let's take a look at what the somatosensory cortex looks like in a normal monkey. Here what we see is that the somatosensory cortex has a beautiful logical organization. On the right, where it says 1D and P, that represents the thumb. D is the distal tip of the thumb, and P is the proximal part of the thumb that's next to the hand. The regions labeled 2, 3, 4, and 5 are the second, third, fourth, and fifth fingers. You see that in the somatosensory cortex, each finger is exactly where it's supposed to be. The second finger is immediately between the thumb and the middle finger, just like it is on the hand. Now, what happens when a monkey trains intensively on this task? Well, here, what we see is that there's dramatic reorganization of the somatosensory cortex. The regions labeled 2D and 3D, which represent the distal tips of the second and third finger, are now much, much larger. Now that's astonishing because what it means is that in the adult monkey, the somatosensory cortex has completely reorganized to represent what's important to that monkey, which is now input to the tips of the second and third finger. And this was really the first demonstration that this kind of functional wiring change could occur in an adult brain. Next, researchers in Dr. Mahncke's lab, this time led by Dr. Greg Reckenzone, showed that this kind of brain plasticity was also the neural basis for learning. Now, to show this, they had to have a monkey learn something. And in this case, the monkey learned to do a tactile task. He puts his hand out into a hand mold, and in the hand mold, there's a vibrotactile stimulator. And what this does is it delivers a short, soft vibration to the monkey's finger. The monkey essentially feels a little vibration, and it goes like bzzzt, bzzzt, bzzzt. And then there's one that's faster, bzzzt. And what the monkey learns is that when he feels the faster one, if he pulls his hand out, he'll get a little reward. Now, when monkeys do this at first, they're not very good at it. That faster one has to be much faster for them to notice it. But as they train day after day for a few weeks, they get very good at it. They can detect a stimulus that's only a little bit faster than the one that came before it. So what the researchers asked was, well, what has changed in the brain to allow a monkey to do this task? So now what we're looking at here is recordings of electrical activity in the somatosensory cortex. First, what we're seeing is the electrical response to that vibrotactile stimulator on the finger that the monkey has practiced at. And what we see is that the electrical activity shown in black here is very, very closely matched to the exact vibration pattern that's coming onto the monkey's skin. And what that means is that this monkey has fast, accurate information processing, and as a result can do this task in a fast, accurate way. But now if we looked at the electrical response to that same vibration delivered to an adjacent finger where the monkey hasn't practiced or learned the task, we see an electrical response that scientists technically call schmutz. All of this noisy response here, you can see, does not represent the vibrotactile stimulation very precisely. And in fact, it's because the monkey's brain information processing is noisy here that as a result, the monkey is not very good at the task. So this result was a breakthrough because what it told us was that training the brain could make brain information processing faster and more accurate, and in fact, making brain information processing faster and more accurate could completely explain the magnitude of learning, in this case, that a monkey has done. Researchers then went on to show that the changes that we can drive in brain function are incredibly specific. This work was again led in Dr. Michael Mahncke's lab, in this case by Dr. Dan Polly. What we're looking at now is rat brains, and particularly the auditory system of rat brains. Here, in a control rat, what we see is that the auditory part of the brain is laid out in a very well-organized way, and in fact, much like piano keys are laid out. In blue, these represent regions of the brain that respond to low tones. Yellow and orange respond to middle tones, and red responds to high tones. Now, what the researchers did was they designed a task where a rat would hear a series of tones, and then if she heard a very specific frequency, in this case 4 kilohertz, she was taught that she could run over and get a quick food treat. Now, in a control rat, you can see that only a few regions of the brain, shown in gray, respond to that 4 kilohertz tone. It's not that important to an ordinary rat. Now, if we look here, we look at a rat who's trained to do this task, and there's been this dramatic reorganization. Much more of the brain now responds to that 4 kilohertz tone shown in gray. And that's because, well, responding to a 4 kilohertz tone has become incredibly important to a rat. Now, if you do that task a little bit differently, where now the rat has to notice that if the tone has a certain loudness, that she can go get a food reward, you see that there's actually no frequency change that occurs in the rat auditory cortex, and that's because frequency was not that important to the rat. Conversely, on the bottom, if we look at loudness maps, and here what the light blue shows is regions of the brain that respond to the loudness that a rat might be trained to respond to. Again, in a control rat, there's just a few regions that respond to that loudness. If a rat's trained to respond to frequency, that loudness map doesn't change very much. And here we show a rat who's been trained to respond to a specific loudness, and we see now that there's a number of regions in the brain that respond specifically and only to tones of that loudness. So what this shows us is that the brain dynamically reorganizes itself to exactly what's important to the rat. And what that means is that attention and reward, in fact, gate brain plasticity in this way. Now, attention and reward systems in the brain are referred to as neuromodulatory systems because they broadly and diffusely control the overall kind of activation of the brain. So what was quite interesting is that next, researchers asked, well, hey, are these neuromodulatory systems themselves plastic? Can they be changed by experience? And in beautiful research led here by Kimberly Garrick, what she showed was that in fact that's true. And what she's doing here is she's looking at how much dopamine is expressed, in this case in rat brains, and that's shown on the y-axis. And she looked at 4 different groups of rats. The first group, shown in the white bar, are just ordinary rats living their lives in ordinary rat cages, and they have a specific amount of dopamine on the average in their brains. Next, she looked at a group of rats that she had exposed to a neurotoxin called MPTP, and MPTP is known to specifically damage dopaminergic systems in the brain, and you can see in black it does its job. These rats have much less dopamine. Next, she asked a very interesting question. Could the level of dopamine in the brain of a rat be changed by what the rat does? And she used a very simple intervention, which is an exercise intervention. In this group of rats, shown in dark gray, these rats had about 3 months of intensive exercise, running on wheels and so forth. And what we see right off the bat is that there's more dopamine expressed in these rats who've been exercised. So that gives us the idea right off the bat that in fact neuromodulatory systems, in this case dopaminergic systems, are in fact plastic and can be changed by what a rat does. And then in the most interesting result, she showed that those changes are neuroprotective. If she took rats who had been exercising for 3 months, who presumably had that increased level of dopamine, and she damaged their dopaminergic systems with MPTP, although the dopaminergic systems were still damaged, those rats had significantly more dopamine left than the ones who had not exercised. And what that tells us is that the neuromodulatory systems in the brain are not only plastic, but those plastic changes themselves can make the brain more resilient. So this set of observations from really decades of experiments in brain plasticity in animals as well as in humans led us to a set of principles that govern the design of building efficacious brain training exercises, brain training programs that work. So when we at Posit Science build exercises, we follow these 5 principles. Virtually every exercise that we build first focuses on speed. We want to make the brain faster. And to do that, exercises typically employ increasingly brief stimuli, shorter and shorter sounds, faster and faster visual images that a person has to process and identify. But of course, accuracy is also important. We don't just want the brain to be faster, we want it to be faster and more accurate. And to do that, virtually every exercise employs increasingly challenging discriminations, things that are harder and harder to tell apart that a person can hear or see. Also, these exercises have to be adaptive. And what we mean by that is the difficulty level has to constantly change to match an individual user's performance. The way we do that is as a user gets trials correct, we make the exercise harder. And if a user gets a trial incorrect, we step it back and make it easier. That helps us maintain a level of challenge for each user where they typically get 70 to 90% of trials correct. This is really important for learning because if a task is too easy, if a person's getting 100% of trials correct, there's no need for the brain to change. And if the task is too hard, a person is just guessing, well then the brain doesn't change either because it has no opportunity to start to change. Next, we design the stimulus sets in all of these exercises to drive generalizability. We don't particularly want people to get better at only what they practice at. We want to have a brain that's ready to operate quickly and accurately on the kinds of things that a person encounters in the real world. The way we do that is we begin brain training exercises with strongly emphasized stimuli, slow, loud sounds, bright, sharp objects that pop off of the background. And we do that because it helps strongly drive brain plasticity at first. But as a person works their way through the many levels of each exercise, they gradually move on to complete training with naturalistic stimuli that have statistical properties in the auditory and the visual systems that are similar to what a person encounters in the real world. And this helps efficiently span stimulus dimensions of real-world stimuli so that the brain, after it's gone through training, is pre-adapted, one might say, again, to quickly and accurately operate on the kinds of things that a person's going to hear and see in the real world. Finally, we design these exercises to be engaging. And here, I don't just mean that they're fun, although it's nice that they're fun. What I mean is that these exercises need to frequently and repetitively engage neuromodulatory systems like the cholinergic attention system, dopaminergic reward systems, and novelty systems that depend on noradrenaline and serotonin. We want to activate those systems on and off many, many times over the course of a source of training because it's through that engaging that we believe we can re-engage and renormalize these neuromodulatory systems. So let's put these brain training principles to work. Here we're going to look at a plasticity-based brain training program built for rats. Here we see our rat subject, ready to learn. And in this task, the rat's going to hear a series of tones like boop, 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 beep, boop. The rat's going to learn that every time she hears that oddball, if she pokes her nose in a little sensor, she's going to get a little treat. So of course she rapidly learns how to do that. Now it gets fun. We can then make it faster. We can make the sounds go like boop, 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 beep, boop, boop, boop. And in fact, we can make the oddball less distinct. We can make it so it goes boop, 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 beep, boop, boop, boop. In this way, the rat's brain is trained to become faster and more accurate. So let's see what effect that has on rat brains. This work was led by Dr. Etienne de Villers-Sidani, who was working with Dr. Michael Mahncke at that time. So we're going to look at three groups of rats. Here's a young rat, here's an older rat, and here's an older rat who's been through brain training, and that's why she has that beautiful little hat on. Let's first look at what this does to information processing in the rat brain. That's shown here in this map of neural precision. On these maps, areas of the rat brain that are quite precise, that respond to only one sound, are shown in blue or light green. Areas of the rat brain that are quite noisy, by which I mean they respond to lots of sounds, and so they don't give the rat very good information around what the rat is hearing, are shown in red. And in the young rat, we see that almost all of the auditory cortex is quite precise. But in the older rat, we see more and more regions of yellow, orange, and red, and that means more and more of the rat brain is quite noisy. It's not doing a good job telling the rat what is actually going on in the outside world. And of course, this happens to people as well. This is why older people often say, for example, I can't really follow what my grandkids are saying. They're just talking too fast. Or, when I go to a restaurant, I have a hard time hearing what people are saying and all the background noise. It's because on the average, their brains are like this older rat's brains. There's just more parts of the auditory system that are noisy and imprecise. But if we take a look at this map for a rat who's been through that intensive brain training, about an hour a day for a month or so, we see this fantastic renormalization of precision. And in fact, the brain of the older rat that's been trained is about as sharp and precise, or even maybe slightly more sharp and precise than that of a younger rat. So we can drive dramatic functional changes as a result of brain training in rats. But how about what's changing about the brain itself? Well, in this next column, what we're looking at microscopic images of neurons in the rat brain. Each of those red dots is a specific kind of neuron called a parvalbumin positive inhibitory interneuron. What this network of neurons does is it helps coordinate global brain state, making sure essentially that different parts of the brain stay in sync with each other. Now in young rats, we see quite a few of these kinds of interneurons, but in older rats, we see many, many fewer. And we're not quite sure if they're dying or becoming deactivated, but one way or the other, there's not as many of them functionally present. But in an older rat who's been through brain training, we see something dramatic. We see that substantial renormalization of the number of these kinds of interneurons in the rat brain. There's about a 50% restoration. And that's simply through doing this intensive brain training. One more example of what changes in the brain here is shown in these microscopic images of neural wiring. What the red represents here is the level of myelin Myelin is a substance that essentially insulates the neurons as they connect to each other. You might think of it in the same way that insulation occurs in the wiring in your house. In young rats, we see quite a lot of this myelin in the cerebral cortex, as shown here. But in older rats, that level of myelin has substantially decreased. And in fact, some of that lack of insulation may be what's contributing to noisy information processing in the older rat brain. But then in an older rat who's been through brain training, we again see this nice improvement in the level of myelin, of insulation of the wiring in the rat brain. What this tells us is that this kind of brain training is not only changing the behavioral abilities of the rat, it's not only changing the information processing abilities of the rat, but it's quite literally changing the basic biology of how the brain is wired together. So we've built brain training programs for rats. It improves the rat's performance, makes their brain sharper, and even improves the health of those rat brains. It's probably time to do that for people, and that's what we've done here at Posit Science. We take these basic principles of brain plasticity, like speed, accuracy, adaptivity, generalization, and engagement. We make software-based brain training exercises that people can use. We deliver those brain training exercises to large numbers of people, which gives us a really good idea of, in each exercise, what good performance looks like and what not so good performance looks like. And in fact, then we use adaptive algorithms to help us determine, well, what order should a person work their way through these exercises in to drive optimal performance gains. And that's what BrainHQ is. A set of more than 24 brain training exercises built on the principles of brain plasticity that are designed to rewire how the brain works to make it faster and more accurate. So what can we do with this technology? Well, if we can improve the speed and accuracy of information processing in the brain, if we can re-refine cortical maps, if we can strengthen neuromodulatory function, well, these kinds of brain training exercises could be helpful for many different kinds of people. And in fact, that's what brings us to work every day here at Posit Science, is the basic belief that it's time for this science of brain plasticity to move out of the lab, where we've helped rats and we've helped monkeys, and move it into the real world, where we can help people. So thanks for listening to this webinar today. If you'd like to learn more about the science that I've talked about, here's a set of references. And you can always come to brainhq.com to try out these exercises and learn more about the science. Thanks.